Okay, well, we're dealing with chapter 25 and really just the metabolism of carbohydrates. So pages 977 through 990. Remember, metabolism refers to all chemical reactions in the body, and this is that same old uh, diagram that we've seen more than a couple of times. We start at the top here with simple molecules such as glucose, amino acids, glycerol, or fatty acids, and we can uh, use anabolic reactions and generate complex molecules with those. Um, and with those complex molecules, we can store stuff, store energy, and then release that energy with catabolic reactions to form ATP. So we see ATP here in the center, and how ATP links these catabolic and anabolic reactions back and forth, back and forth. We start out with ADP, adenosine diphosphate, plus free phosphate. Using catabolic reactions, we can then make ATP. ATP can then be used to generate complex molecules in anabolic reactions. Okay, and then those complex molecules can be broken down, and ADP and phosphate generate new ATP. So remember, when we're doing this, or when you're doing this, that we're going from simple to complex, and in that, going that way, we're storing potential energy. So storing potential energy. So that is potential energy. Potential energy is going up. We're storing potential energy. Here, with the catabolic reactions, we're releasing that. We're releasing that into hopefully into kinetic energy at some point. But on this side, potential energy will be going down. I want to make a nice arrow. So potential energy will be going down because we'll be tearing apart these molecules. Here, potential energy goes up because we're storing energy and generating complex molecules. Okay. This energy transfer occurs with a set of reactions we call oxidation reduction. Oxidation or redox reactions transfer hydrogen ions from one molecule to another. So as an example, taken right from the book, you start out with lactic acid, and lactic acid itself is is already reduced, and NADH plus, it's an NAD, nicotinamide adenine dinucleotide, is what NAD stands for, that is already oxidized and is an energy storage molecule. Well, what happens is, that's not even, we don't even really need to know about that. I mean, about the state of them already. We're really concerned with where they're going. And lactic acid will go to pyruvic acid, and NAD will go to NADH. And I'm drawing these arrows kind of curved because these two molecules really are interacting with one another. And when lactic acid goes to pyruvic acid, it is oxidized. Okay, so pyruvic acid is oxidized. NADH plus an extra hydrogen is reduced. What's really happening is, what oxidized really means, I think we mentioned this the other day, is it loses electrons. It's oxidized. So we remember Leo. We lose electrons, we're oxidized. And if we gain electrons, we're reduced. So what's happening is electrons from lactic acid are being shuttled over to NADH, which is picking up those electrons and becoming reduced. It's gaining electrons, so we gain over here, and that is becoming reduced. We lose over here, we're losing that electron over here, so that is becoming oxidized. Okay? And when a substance is oxidized, it means that the molecule is being torn apart. When the molecule is being torn apart, that's really being catabolized. If it's being catabolized, what's happening to potential energy? Potential energy is going down. Here, on this side, we're creating a larger, this is a larger, just look at how many letters we have here. There are more letters, so it's larger. So this, we're creating new and complex molecules, so it's really going through anabolism. So what is happening over here to potential energy? Potential energy on this side is going up. Okay. So if you're being reduced, you're gaining potential energy. If you're being oxidized, you're losing potential energy. An ATP generation, all right, ATP is that molecule that we need, that we store energy on, that we can pass energy off to do kinetic work. Okay, so we can transfer this potential energy to kinetic energy through ATP. And ATP generation really happens in two different ways in humans. There are actually three ways it happens, we'll see. But the first is called substrate level phosphorylation. Basically what we're talking about here is there's some kind of substrate, and usually that substrate is an organic molecule that has a phosphate attached to it. So it is phosphorylated. And we're generating ATP by transferring that high energy phosphate that's on that organic molecule to ADP. Okay? So the phosphate is already attached to something, and it's being reattached to ATP, or to make ATP. That's substrate level phosphorylation, transferring a high energy phosphate from an intermediate. That intermediate is going to be an organic molecule directly to ADP. There's also oxidative phosphorylation. Now, this is, remember, phosphorylation, this word, phosphorylation, is any, or adding a phosphate to something. So a molecule has been phosphorylated if it has a phosphate added to it. Here, substrate level phosphorylation, it's coming from a substrate. The phosphate is coming from a substrate. Here, with oxidative phosphorylation, it occurs in the presence of oxygen. That's where we get oxidative. So what we do is we remove electrons from these energy molecules, the food that we eat, and they pass through an electron transport chain ultimately to oxygen. When these electrons pass through an electron transport chain, they actually generate energy, and that energy is used to attach a free phosphate to ADP. So instead of using an intermediate, we attach a free phosphate to ADP to make ATP. And lastly, there's photophosphorylation. This only happens in plants. It's phosphorylating ADP to ATP, really in the presence of photons or light. Okay. There are a bunch of fates of carbohydrates, and it really depends on what the body needs. If the body needs energy, carbohydrates will be reduced to, or oxidized, pardon me, carbohydrates will be oxidized to make ATP. If ATP really isn't needed, then carbohydrates can be used to do a number of other things. It can be used to make amino acids, some amino acids. It can be used to make glycogen. Remember, glycogen was also called animal starch, and carbohydrates could be used to make triglycerides, which of course is fat. ATP production itself is a very involved process. I'm going to be going over some of the main points to it. It's obviously very, very complex, as you have read in your chapter. And we'll go into a lot of the complexity when we're in class. Okay, so we start out with really a four phase. We're going to talk about phases and steps. 4 phases and multiple steps within each phase. So the first phase over here is glycolysis. Glycolysis is just what it says. The lysis, the breaking of glucose. So we start out with one glucose molecule and we break it. We break it to 2 pyruvic acids. Okay? And when we do that, this is kind of a net effect here. We get out 2 ATP, so that's what we want. We want ATP, but we also generate 2 NADHs plus a couple of hydrogen ions. NADHs is another nicotinamide adenine dinucleotide with a hydrogen, is another energy storage molecule. Remember we said like ATP was our AAA battery. That's what we need. NADH is kind of like still like a D cell battery or a C cell battery. It's too big. So we're going to have to ratchet that down to actually make ATP. So NADH really is just energy storage that we'll use later. So it's something to keep in mind. It's there, but we're not going to do anything with it yet. Okay, so there's phase 1, glycolysis. Phase 2 is getting stuff ready to go on to make a whole bunch of ATP, and this phase is the formation of a little molecule called acetyl coenzyme A, or acetyl-CoA. So we take 2 pyruvic acid and we make 2 acetyl-CoAs. When we do that, you'll see right here, off comes some more NADH, which is great. We'll use that for energy later, but also 2 carbon dioxide molecules. Now, if you remember, glucose itself, the chemical formula C6H12O6. Okay, so we have a 6 carbon molecule, and that molecule is actually organically a hex ring. 1, 2, 3, 4. Oh, this is always so difficult. 5, 6. 1, 2, 3, 4, 5, 6. Okay, and then so there's carbon number 1, 2, 3, 4, 5, and then I'm running out of space here. Up there is 6. So there we have our 6 carbons. It's actually supposed to be standing up a little bit more. There's oxygen there at that corner. Okay, maybe I should redraw this. We start out with oxygen. It's still too high. Nerds. Carbon, 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 carbon. Okay, 1, 2, 3, 4, 5, and then the 6th one up there. Oh, I could have made it the C from process. Okay, so what we've done is we've now taken this hex molecule and we've actually made 2 3-carbon chains. Okay, that's pyruvate or pyruvic acid. So we've taken this and broken it down into these 2 3-carbon chains, and now when we go to acetyl-CoA, we actually get rid of a carbon there and a carbon there, so we're left with a 2-carbon molecule. And these two carbon dioxides we breathe out. Okay. Once we've made acetyl-CoA, it's gone into the mitochondria. The mitochondria, remember, the powerhouse of the cell. These two tiny molecules then go into the Krebs cycle. Krebs cycle is a cycle, or it's also called the citric acid cycle, because the beginning product, the beginning part of the whole cycle starts with citric acid. It runs through this cycle, okay, and in doing so, take a look, we get out 2 ATP, 4 more carbon dioxides, a whole bunch more energy storage of NADH, and more energy storage in another molecule called FADH. Now this is really great. Okay, so we've gotten rid of 2 carbons there, so we cross those off. 1, 1, 2, and then look at that. 4 more carbons come off. 1, 2, 3, 4. So by the time we're done with Krebs cycle, all of the carbons from our original glucose molecule have been converted to carbon dioxide, and we'll breathe those out. Okay, so that's phase 1, 2. Krebs cycle is phase 3. And then finally, the electron transport chain is phase 4. This is where most ATP are made right here. Electrons are passed through this transport chain, and the electrons come from our NADHs that we made and FADH that we've also made, and those electrons are passed down, high energy electrons, passed very carefully like a hot potato down this line, and at the very end the electrons are accepted by oxygen, and we make water out of it, and in doing so, energy passed, we'll see how this all works. We can then make 32 or 34, theoretically, 32 or 34 more ATP. So we've gone from 2 up in glycolysis, 2 in Krebs cycle, all of a sudden 32 to 34. So that's what we're shooting for. Okay. Glycolysis, in the first phase, is a 10-step process. It won't take you very long to memorize all 10 steps, and I don't know why you'd want to do that, because it's kind of silly. But what you should note is that, okay, so this is a very involved process, and in other words, it takes a lot to take a glucose molecule and break it apart into 2 pyruvic acid molecules. It's not an easy task. And because it's not an easy task, one of the first things you'll notice is, take a look at that right there. First step, we have our glucose. First step, we phosphorylate glucose. We take ATP, and a phosphate comes off, and it goes to ADP, and now we have glucose 6-phosphate. In other words, we've chunked on a phosphate out here onto carbon number 6. Okay, what has that done? Well, I've done an oxidation reduction. I have made this molecule I've made bigger. Okay, so it's gone through anabolism. So what's happening to the potential energy in this molecule? Well, the potential energy in ATP has gone down. It's lost. Okay, potential energy in this molecule, we'll just call it PE, the potential energy here has gone up. Okay, hopefully, and ultimately what it's doing is it's starting to make this molecule unstable. Glucose isn't a molecule, it's just going to fall apart on its own. It needs to be primed to fall apart. It's kind of like that old, you can't make an omelet without breaking eggs. Here, we can't make energy without spending energy. So maybe it's more like you can't make money without spending money. Here, you can't make energy without spending energy. So we phosphorylate this glucose, and take a look what happens. It's phosphorylated, and all of a sudden we've gone from a hexagon ring to a pentagon ring. So we've shifted this molecule around, and made a new molecule, a fructose 6-phosphate molecule. So we've gone from glucose to fructose 6-phosphate. And then take a look what happens again. We add another phosphate. We phosphorylate this molecule again. And so now it's fructose 1,6. So that's telling you phosphate out on 1, phosphate out on 6. Okay? So now it's fructose 1,6 bisphosphate. That, again, what does that do to potential energy? Oh, yeah, you guessed it. Potential energy goes up again because we've made this molecule just a little bit bigger. That makes this molecule even more unstable, which is what we want. And then the molecule breaks. So here we have it. This molecule finally breaks. Now we have 3 carbon molecules. Count them. 1, 2, 3. 1, 2, 3. And these arrows are noting that it goes back and forth between glyceraldehyde 3-phosphate and dihydroxyacetone phosphate go back and forth. They're just interchangeable, sort of. Okay, so we have this. We finally got to our 2 3-carbon molecules. Now we can start to tear these 2 molecules apart. When we do that, what are we going to do? We're transferring energy over from these molecules to energy storage. And there's our first one, 2 NADHs. And then here's our next one, 2 ATPs. We finally made ATP. But remember, we had to use 2. Now we make 2. So how many ATPs do we have right now? Net 0. We're at ATP equals 0. Okay? Because we used 2, made 2, ATP's at 0. We were down 2. So let's say at this point ATP minus 2. We've used 2. Now we're at 0. Now we continue to rearrange this molecule, tear it apart, and there are 2 more. So now we're at ATP equals plus 2. Okay. And then we've wind up at the very end here with 2 pyruvic acid molecules. If oxygen is present, we continue. If oxygen is not present, we're done. Let's go back to this. If oxygen is not present, we're done. We've made 2 pyruvic acids. We can't go any further. And in fact, what will happen is we will take these pyruvic acids and convert them to lactate or lactic acid. And to do that, we actually have to take these 2 NADHs that we just made and use them here. So this takes energy to convert pyruvate to lactate. And when we do that, we use those energies, that stored energy. So if we're doing anaerobic without oxygen, we ultimately make 2 ATP, and that's it. No other energy storage, nothing. Okay, because we use our other energy storage to make lactate. If oxygen is present, we continue. Okay, so there was phase 1. We're down to pyruvic acid, and then we convert. There's acetyl-CoA right there. So there's phase 2, and in doing so, and when we convert that to acetyl-CoA, carbon dioxide comes off, and we make a little bit more. Since we've lost energy on these molecules, we store it there. Okay. Now, here's Krebs cycle, and you can see why it's a cycle, because we start out here at step 1. Acetyl-CoA comes in, and it combines with citric acid, or oxaloacetic acid, and makes citric acid. So this is why we sometimes call this the citric acid cycle, because the first step we make citric acid, and then we go through kind of like we did with glycolysis. There's a series of steps, 8 steps here, and as we're going through, all we're doing is shifting the molecule around, and every time, when we shift the molecule around, we can get carbon dioxide to come off. That lets us capture some energy there, and we have carbon dioxide come off again, and we capture some more energy there. Now what's really important is, remember how many CoAs came in. There were really two molecules coming here. So right here there are 2 and 2. So there's 2 and 4. That's all of our carbon dioxide. We then go through some other shifting around, and we capture FADH and NADH. Those are more energy storage. Those will all go to the electron transport, our next step. And then way down here, kind of buried, you can see that GTP, another energy storage molecule, and just an intermediate, creates more ATP down here. So there's 2 there. Okay. We talked about substrate level phosphorylation. That happens in glycolysis. That also happens in the cytoplasm. Okay. Oxidative phosphorylation occurs in the presence of oxygen and occurs in the mitochondria. Oxidative phosphorylation happens through this process called chemiosmosis. This is just so cool. Remember we made NADH and FADH. NADH, here it is. It's bringing these electrons over here, and it passes off these electrons, so it's lost an electron. It's reduced back down to NADH, and the electron is passed to this electron transport chain. And you can look in your book on page 986. It has all of the electron carriers that are present. There are a ton of them in here, and they're protein and non-protein molecules that serve as carriers. But what's happening is these electrons are being passed very carefully, because electrons by themselves can be very dangerous. If you don't believe me, go stick your finger in a light socket. Those are free electrons. They kind of hurt, and they could tear a cell apart. But as they're being passed, what's happening is, check this out, NADH plus H goes to NAD. That leaves a couple of H's lying around over here. Well, what happens is, as electrons are getting passed, those H's are being pumped across the membrane. So if you don't remember how the mitochondria is set up, you may want to take a look at that real quick. There's a mitochondrial matrix, the very inner part, and then there's the inner membrane space. So these hydrogen ions are being pumped through these pumps, these proton pumps, and through here, through these proton pumps, and we're setting up a whole bunch of hydrogen ions on this side in this inner membrane space. And hydrogen ions cannot get through the cell membrane or through the mitochondrial membrane because they're ions. Okay, so they build up on this one side. Okay, this is part of chemiosmosis. Okay, and oxidative phosphorylation. What happens with all of these hydrogen ions is they can get through. There's a hydrogen channel over here, but this hydrogen channel happens to be in part of this molecule called ATP synthase. And when these hydrogen ions pass through ATP synthase, it spins this molecule like a huge turbine. And that's, what kind of energy is that? Oh, it's kinetic energy, energy of motion. So that kinetic energy is used to take ADP in a phosphate and make ATP. And these electrons, these very dangerous electrons, are finally passed to half of an oxygen molecule, which is an O, but we can't draw it just O. We have to say half, 1/2 of an O2, okay, and makes metabolic water, H2O. Okay, so all of these electrons used to pump hydrogen ions across. Hydrogen ions fall back through ATP synthase. That generates more ATP. The electrons are gingerly passed through all of these electron carriers to oxygen and a hydrogen ion bind up, a couple of hydrogen ions bind up with those and make water. Okay, so I know this picture is on its side, but I wanted to leave it big. So glycolysis itself, which is down here, glycolysis generates only 2 ATP. While using them, and if remember, if without oxygen we stop there, so that's it. Anaerobic would generate 2 ATP. When we use the mitochondria and use Krebs cycle within its intermediate acetyl-CoA, Krebs cycle, and then the electron transport chain, we can make theoretically up to 38. A lot more. And finally, we talked about some of the other fates of glucose. Glucose can be stored as glycogen by the liver, and we take blood glucose, and here's our steps. Blood glucose comes out. We go through these series of steps. It actually takes energy to do this, because we're building a complex molecule, so it's going to take energy. We build it up, make glycogen. Glycogen is animal starch and stored in the liver. And this is glycogenesis. Glycogenesis, the formation of glycogen stimulated by insulin. If this needs to come back out, it's not the same steps. We actually skip this step in here, but then finally get back out to blood glucose. When the body needs more glucose in the blood, and that's stimulated by glucagon, another hormone, and epinephrine. Okay. The body can also create glucose from proteins and fats if it needs to. It can take lactic acid or certain amino acids, go into pyruvic acid, convert those to pyruvic acid, and then push that back up and make glucose out of it. Can also use glycerol from fat and push that in here and get glucose back out. And this is called gluconeogenesis, stimulated by the hormone cortisol and also glucagon. When your body needs glucose, it will do this. The body can also create fats and store sugar. If you take an excess sugar, it will store it as fat. So it can go the other way. And there's my cat.